Hello, viewers. Welcome to the ninth question answer session. In this session, we've got only one question, and that is about docking. So let's begin. Right, the question has been asked by Shima, and she asks, what is docking and which type of natural products are appropriate for docking and or to perform molecular docking. So it's a very generalized question. First of all, let's see what docking means. So molecular docking is a computer-based molecular biology tool that predicts the interaction between a ligand and a receptor. The ligand, in this case, can be a molecule, a small molecule, and the receptor is the protein. So it actually predicts the interaction between a ligand, a molecule, and a protein. It basically predicts the preferred orientation of the ligand for binding with the active site of the receptor or the protein. Now, the concept of molecular docking started back in 1980s, and since then it has developed into a very useful tool, especially for drug design and drug development. So over the past four and a half decades, molecular docking techniques have evolved so much that it can now actually help scientists in drug development, so much so that some of the drugs developed or designed through molecular docking are already in the market. Now, what molecular docking is? So if you consider a molecule, and here we have a protein has its active site. This molecule comes and it interacts with the active site, the groups in the active site of the receptor of the protein. So this binding actually is predicted by molecular docking or through molecular docking, and it actually predicts the preferred orientation of the molecule, in which orientation this ligand is best fit for binding with this receptor. It helps in calculating the binding energies, which actually then helps in deciding which ligand is more fit for binding with a particular receptor. Now, as you have asked about natural products, so we can actually screen any compound, any type of compound, whether that is a natural compound or a synthetic compound, and test it in silico through molecular modeling and molecular docking, and evaluates its efficacy or its binding affinity with a particular type of protein. Now, the question you have asked is more generalized. Like you have asked about which natural products. Now, actually, there are two types of approaches in molecular docking. We need to understand that one is a target-based approach and the other is a ligand-based approach. So both the strategies can be used for predicting or for designing lead molecules. Now, because we are more interested in drug development or drug design. Drug development, it's a very long process. It takes about 14 to 15 years for a drug to come out of the lab and land into the market. It's a very lengthy process which includes the discovery or the synthesis of a particular molecule, its in vitro testing, in vivo biological evaluation, then preclinical trials, clinical trials, and then it needs an approval from the FDA. And so it takes a lot of time. And it has been reported that the cost for a single drug development has increased by about 145%. Although the time for a drug to come out of the lab and land into the market, it has reduced. But along with that, the percentage of rejection by the FDA has also increased many folds. Now, I'll tell you how molecular docking can help us reduce this time frame and be more specific while designing or developing lead molecules and ultimately a new drug. So as I told you earlier, that the drug development, it has different steps, starting from chemistry lab, where the compound is discovered or it is synthesized, and then it is evaluated through different bioassays. Then it goes into in vivo testing, preclinical, clinical trials, and so on. So what molecular docking does is that it's actually a computer-aided drug design. So what we do is that we screen a large number of molecules. For example, 500, 1,000, 5,000, 10,000 molecules are designed theoretically, and then they are screened against a particular enzyme, a particular receptor. Now, why it is important to screen a particular molecule against an enzyme? It is very important to know that enzymes or proteins are involved in the onset of a particular disease. For example, let's say urease enzyme, it is involved in the onset of peptic ulcers. So in order to treat peptic ulcer or to prevent peptic ulcer, it is very important to inhibit the urease enzyme. So how would we know that what sort of molecules would be good enough to inhibit this particular enzyme? So again, what we do is that we design a large number of molecules, a library of molecules, and then screen them against this urease enzyme in silico on the computer screen. So we have the receptor and we have the ligands. These ligands, these small molecules which have been designed theoretically, they may have similar skeletons with different functionalities, or they may have different skeletons altogether. So you can design different types of molecules, and then we screen them against this urease enzyme. We calculate the binding affinities, the binding energies, and then we decide which of these ligands, which of these molecules they can best fit or bind with this urease enzyme and inhibit its activity. Now, out of those molecules, the active molecules are selected, and then you can actually go to the lab and synthesize those molecules. So you will have a picture in your mind what sort of molecules you want to synthesize to inhibit the urease enzyme. In fact, you will have the actual structure of that molecule. Now, this molecular docking strategy, it's still, despite development over the last four and a half decades, is still far from perfect. So you actually still have to do some modifications or optimizations in the structure to get that particular lead molecule which is effective against a particular enzyme, and like for example against the urease enzyme. So you can actually perform the structure activity relationships, the SAR studies, the QSAR studies can be performed using this molecular docking technique. This is done with the help of different softwares which are developed over time. Some of them are the Autodoc Vina, Autodoc Gold, and the MOE software, FlexX. These are some of the softwares that you can use for in silico or molecular docking studies. Now, some of the softwares are like, none of the software may be effective in all studies. So you actually have to select suitable software for your according to your requirements. So in short, molecular docking is a technique that can help in drug design, in drug discovery, and drug development, and especially for chemistry students, for organic chemistry, for biochemistry students. It's very important to learn this technique, to help to learn how to use it, to utilize it to the fullest, in especially in medicinal chemistry or in drug discovery process or in drug designing process. And it still is a pretty helpful tool, but again, you have to go to the wet lab and perform real-time experiments to actually evaluate a molecule and confirm its efficacy against a particular disease. So I hope your question has been answered. If you still have any questions regarding the same topic, feel free to ask, and we'll have another session on that too. For rest of the viewers, do send your questions on the email address given below. I'll be waiting for your questions. See you next Friday with new questions and new knowledge. Thank you so much for watching.